Good morning to everybody. I would like to thank you for giving me this opportunity to present here my data. I will speak about the functional evaluation of immortalized porcine endothelial cells in a whole blood clotting assay. Why porcine immortalized endothelial cells? Well, because primary endothelial cells, as we know, has a limited lifespan in culture. We can culture them after several passages they reach a state of replicative senescence, so they stop dividing and you cannot use them anymore. They will have changes in cell morphology, gene expression, and metabolism, and because there is the activation of tumor suppressor proteins like P53, RB, and P16. What are the advantages and disadvantages of using immortalized endothelial cells? By using them, you can overcome senescence, you can have a rapid growth of the cells, and you can have minimal care of them because they are very strong cell lines. You can culture them for several passages, unlimited growth, and reduced number of isolations. The disadvantages could be that they could have different behavior compared to primary endothelial cells, and uncontrolled growth, because you can have that they can grow one on top of each other. How is the immortalization process? These cells are derived from porcine aorta. They are endothelial cells, and they are transfected by SV40 viral antigen. And after a drug selection, you can have the immortalized cell line that's called PED, and they became from wild-type porcine aortic endothelial cells. From this cell line, we obtained, after transfection by electroporation with a targeting vector, the alpha-gal knockout phenotype. So this work aims to answer these questions. Do porcine aortic immortalized endothelial cells retain the same endothelial cell phenotype even after the immortalization process? Is the anticoagulant environment provided by these cells as primary porcine endothelial cells do? Because the idea is to evaluate the possibility to replace the primary endothelial cells with these immortalized cell lines in our in vitro assays if the two above conditions are verified. But before using them for this assay, it's useful to do a phenotypical and functional studies before using them. So what I did is first immunofluorescence characterization. This is a staining of the two cell lines, the gal knockout and the wild-type immortalized endothelial cells for the typical endothelial cell markers, CD31, von Willebrand factor, and V. cadherin. And as we can see, they still retain this endothelial cells phenotype. I confirmed this with the flow cytometry analysis. And then I checked the alpha-gal expression of these cells to see whether the alpha-gal knockout immortalized cells are alpha-gal knockout. So what I saw is that they are really alpha-gal knockout. And this is just a comparison with the primary wild-type and gal knockout endothelial cells. Then I did a cell ELISA assay, incubating these cells with the normal human serum. And what I found is that the alpha-gal phenotype of these immortalized cells behave just has the alpha-gal phenotype of primary endothelial cells, because we can see that there is a lower IgM deposition that is in line with the alpha-gal phenotype. But I didn't see any IgG deposition, that IgG deposition is not influenced. I checked also for other complement protein deposition, for example C3b and C4c, C1q for the classical pathway, and I didn't see any significant changes. Then I tried to see whether these cells behave as a primary endothelial cells, so they should carry an anticoagulant phenotype in a physiological situation. This is the model that my colleague Anjan described before. After culturing the cells in the flask, you can transfer there after 1 week in a spinner flask in incubating them with the collagen-coated microbeads. This is a staining of the beads, where you can see that endothelial cells are covering these microbeads. Then you incubate them with the whole non-anticoagulated human blood, and then you take the time until the blood clots. What I saw is that there is no significant difference between the immortalized cell lines compared to the wild type and gall-knockout primary endothelial cells. So these cells behave as the primary endothelial cells, and they delay the blood clotting. In conclusion, PET cells express typical endothelial markers even after the immortalization process. The IgM deposition is lower on PET gall-knockout cells compared to wild-type PIK and PED normal. IgG deposition is not influenced, and the PED cells incubated with whole non-anticoagulated human blood prolong the clotting time of blood. So these cells could be used for our in vitro assay. I would like to thank all the collaborators, Robert Reben, that is my supervisor, and all the other collaborators. Thanks for your attention.
This paper is now open for discussion. Are there any questions?
I have a suggestion.
Yeah.
For your measurement of IgG deposition,
Yeah.
To try different dilutions of human serum, because the human serum contains very high concentrations of anti-Gal IgGs, and you might be at saturation. If you decrease the concentration, you might be able to see the difference.
Okay, of course. Thank you.
Is
Yes.
there a way to do the clotting assay in a high throughput way?
Sorry, I didn't get your question.
Yeah, so I see the whole blood
Yeah.
clotting assay is 1 tube by 1 tube. So can you describe whether there's a way to scale up the assay or have some kind of selection or high throughput screening?
We did a say for example, we take 100 ml of blood, freshly drawn blood, and we incubate them in a ratio 1 to 4. 2 ml of bleeds and 8 ml of blood. And we can repeat this for three tubes each time. So we can have more measurements in the same assay.
For example, if I have 100 different cells, is there a way to easily scale up this assay?
I don't know, actually.
I've got a comment. Congratulations. I'm absolutely amazed. I would have guessed exactly the opposite. I would have guessed that you immortalized these cells and you would have had a disaster. So thank you.
Thank you.
And also, it's great that you keep von Willebrand factor expression in those cells.
Yeah.
Because it's hard to keep in aortic endothelial cells, especially after immortalization.
Yeah. I repeated this staining also after several passages, and they still retain this expression. Yeah.
Yeah.
And to answer the question that maybe your colleague did before to my colleague regarding the tissue factor, I did the staining before, during, and after this whole blood assay, and I saw that there is a sensible increase in tissue factor expression, but the baseline is not expressed.
Yeah.
It's true that you have to be careful when you culture the endothelial cells because they can easily be activated.
No. Well, I mean, that led to a bunch of what I think are erroneous results where people were just describing all the procoagulant activities of endothelial cells. That was a culture phenomenon.
Yeah.
I mean, if you kept the endothelium happy, which is not necessarily easy to do, it would smile at you and be quite compatible with the blood. Otherwise, look like an activated platelet.
Yeah, I agree with you. Thank you.
Thank you very much.